PhD in particle physics from CISA in Trieste. In 2005, he studied high energy physics. After his postdoctoral studies at the School of Physics and Astronomy in Tel Aviv University, Luca joined Stony Brook as a research assistant professor in 2008, and he stayed there until 2017. During that time, he switched into computational neuroscience, that is from high energy physics to high energy brain dynamics. He then spent a year at the Center for Theoretical Neuroscience at Columbia before moving in 2018 to University of Oregon to establish his theoretical neuroscience group in the Institute of Neuroscience. Luca's research focuses on uncovering how coordinated activity of neural populations gives rise to behavior. To achieve his goals, he develops theoretical models of spiking networks and devises novel statistical analyses that combine his expertise in theoretical physics and neuroscience. He's been focusing quite a bit on attractor dynamics and multi-stable states. Rigor and innovation are strong hallmarks of Luca's approach, as you will see in his talk today. I'm very interested to see his talk where he's going to talk about the dynamics of arranging sequences of attractors and the timing of those attractors over a range of timescales. So without further ado, let me first say before I hand it over to Luca, that this meeting is being recorded and streamed on YouTube. If you have questions, please post your questions for the speaker in the question and answers box at the bottom of the screen. It's activated by clicking on Ask a Question. You may also signal your willingness to ask a question in person by ending your question with the words "on screen" in quotes. Okay, and please, you have the opportunity to vote for each other's questions throughout the talk, so the host can know the priority of the questions they might put to our speaker. So Luca, I turn it over to you now. Welcome.
Hi, thanks so much for inviting me. I'm very excited to talk about this research today with you. And so I'll try to share my screen and see if this works. Okay, so I'm sharing the screen, and you should see my presentation. Do you? No. Wait. Do you see? Does anybody? No.
Yes, I see it. Yes.
So you see the presentation? Yes. Is this on? Yes, everything's good. This is the first time I use Crowdcast. So,
yes, we're good. Yes, we're good.
Okay. So yeah, so I'm so excited to be here. Even though I would have rather been here in person. I used to live in New York for a long time, and I used to come to NYU sometimes. So I really look forward to coming back in person. But this is great. So far it's been phenomenal with the Journal Club on the paper I'm going to talk about by Emergence. So that was a great presentation. And so. Yeah, so today I want to talk to you about kind of the leitmotif of the research in my lab. I started my lab 2 years ago in the University of Oregon, and we've been focusing on studying temporal dynamics in neural circuits and behavior. And so I'll give you an idea of three of the first 3 projects that we have in the lab, among many other projects. So. But before I start, I want to talk about the people who did the work that I'll present. And so this include, so do you see my pointer on the screen?
Yes, it's fine.
Yes, awesome. So on the left, you can see people in the lab who contributed to the work I'm going to talk about is Stefano Recanatesi. He's a postdoc. Shared with the University of Washington, Eric Chebron lab, and David Wyrick and Niku Istrater, 2 graduate students in the lab. And Marav Stern was a visiting faculty with us, and she's at Hebrew University now. And these are other people in the lab. And so the work I'm going to talk about today includes these 4 people on the left, and then an amazing collaboration with Ulysses Pereira, who's at NYU. Now he's in Xiaojing Wang's lab. And an experimental collaborators, including Zach Maynan's lab from Champalimaud, and Masa Murakami and Fanny Kazet, who were, Masa Murakami was a postdoc with Zach Maynan, now he's at Yamanashi in Japan, and Fanny Kazet, who's a postdoc with Zach. And if I have time, I'll just plug some of the work we're doing with Rusbe, who unfortunately couldn't make it today. So these are the people are gonna who did the work, and so let me introduce by giving you some kind of general motivation for the work we do. So what we think about are different timescales that appear in animals' behavior. And so we spend a lot of time thinking about how animals behave in the wild. And for now, during COVID, for us, this is like YouTube. And so by looking at the behavior of spontaneous behavior of animals, we can come up with a few hypotheses or a few interesting questions about what temporal structures appear. And so the first timescale that you can see in behavior is the timescale of single movements at a very fast sub-second scale. So these are we call syllables of behavior, and these are the single actions the animal is performing at any given time. So this could be whisking, this could be moving the snout, this could be moving their ears, moving their paw. Every single action, these are very fast actions in the case of a mouse, a rodent that we're going to focus on in the talk. So from these kind of very fast sub-second scale actions, there's an emergent timescale that corresponds to concatenating the syllables into meaningful words. So the animal has a large possibility of concatenating single actions, but they choose to do very simple stereotype sequences of action on the second scale. For example, the animal is walking out of the hole, grabbing the walnut, and carrying it back to the hole. So this whole sequence takes maybe 2 or 3 seconds. And as you can see from this video, they repeat it over and over again. So these are 2 timescales present in behavior. But then there's a third timescale, if you like, that's much, much longer, and that corresponds to the goal that the animal is trying to achieve. In this case, the goal is carrying the walnut inside the hole. And so the animal might repeat this sequence of action of getting out, grabbing a walnut, bringing it back several times over many minutes. So from the simple analysis of this video, or by just playing with your cat or your dog in the park, or just watching animals in the wild, you can realize already that there's a huge hierarchy of timescales in behavior that are occurring simultaneously. So here we see that already we have something like 2 orders of magnitude or 3 orders of magnitude of timescales in behavior. And what we think about in our lab is a way to generate or explain the emergence of this behavior from the point of view of brain circuits. Even though we know that this is a very complex relationship because there's a backdrop of behavior in neural activity, and so we're trying to find ways to simplify setups to extricate this dependence. And so specifically, the questions I'm going to focus on in this talk are the following questions. So how do we concatenate actions into sequences of actions, some behaviorally meaningful sequences of actions? How we can control the speed at which these actions and these sequences are performed once we have a behavioral sequence? How can we control the speed of the reaction times, if you want, the neural responses, depending on the context? And then how can we patch together this hierarchy of timescales within a single neural circuit? So I hope I'll give you in the 3 parts of the talk. I hope I'll be able to to give you some of the possible ideas going towards answering these complicated questions. And I want to start from behavior as a motivation. So this is kind of a seminal work that motivated a lot of the things we talk about. This is work from the Bob Data Lab. They wrote a series of paper where they analyze spontaneous behavior in rodents. And the way they go about doing this is they analyze videos, 3D videos of the animal in their cages for hours and hours, and they process the video, they extract the PCA, and then they fit generative models to these videos of the behavior to extract the behavioral syllables at a sub-second scale. So for example, you can see that the animal performs many different kinds of movements. A rear pause, this is the first one where the animal is moving up, a dive where the animal is diving down, or some ballistic motion in a straight line, that's this locomotion. And so these are color-coded as little segments here during some time course or some spontaneous bout. And so there's many syllables that can occur at the same time. And so the first source of variability in behavior is the large variety of actions or syllables that the animal can perform at any given time. That's the first part of the variability, but then in principle, the animal can combine every action into an exponentially large number of possible action sequences, right? So in principle, some kind of combinatorial arrangement. So the lexical variability in this language, that's nature of behavior, is extremely large. So not only that, but there's another component in variability, which is the temporal variability is also extremely large. So every time the animal is performing one action, for example, these blue intervals here where the animal is performing a dive, they can take however different interval of time to perform that action. They can do it slow, they can do it fast. And so there's a huge variability in the temporal aspects of this behavior. So it seems like that this is kind of a formidable problem, like a very hard problem to crack, how to study the generation, the neural circuits generating this behavior. And so what we thought about doing was trying to find a way to control for this incredible lexical variability in behavior, the fact that you can have many actions and that you can combine them in any possible sequence. And so what we try to do is to start from the simplest part of the variability, which is the variability in the duration of actions. And so what we did is with Zach Main and Masa Murakami, we came up with this idea of, well, they came up with the idea of running this experiment in which they trained rats on a task that had a fixed set of actions or syllables. And the animal was performing that in a fixed sequence, in a fixed order, so just one word. But the animal could perform every single action whenever they wanted. So we control for the lexical variability, and we allow the animal to have self-initiated, spontaneous action initiation within this particular sequence. And so hopefully I'll show you that this allowed us to make a little bit of progress in this direction. And this is the first part of the talk where I'm going to talk about the work done by Stefano and Ulysses. This is the amazing team of theorists that I've been blessed with. And together with Zach and Masa at Champalimaud. This is on the archive. And Menjan just presented this paper like an hour ago. So it was an awesome discussion. So this is the rodent version of the marshmallow experiment that you might be familiar with from psychology. And so the marshmallow experiment, where a kid is left in a room with a marshmallow in front of them, and they have the option of either eating the marshmallow now, or if they can resist and not eat the marshmallow, then they can have 2 marshmallows later. And so Zach and Masa came up with this intertemporal weighting task. And I'll walk you through the task. It's a very simple task. So this is a freely moving rodent in a cage, a rat in a cage. And so there's 2 ports, and the animal can decide they want to engage in the trial by poking into a weight port. That's this the first port on the left here. And then as soon as they poke into the waiting port, a first tone comes on, signaling that's the beginning of the trial. And at that point, the animal has two options, like the kid in the marshmallow experiment. They can either wait for a second tone that's played at a random time in a long interval, and then they can, after they hear the second tone, they can poke out, move to a second port, that's the water port, and poke in the water port and get a large water reward. So this is the patient kid that gets, doesn't eat the marshmallow, so they get 2 marshmallows. But in any trial, the animal has a second option in which they can just poke out anytime without hearing the second tone, and they can still move to the water port, and then they're going to collect a slightly smaller reward. So there's two contingencies, patient and impatient trial. However, the important thing to keep in mind is that the sequence of actions the animal is performing in any trial is fixed. It's always the same in both conditions. So it's poke in, wait, poke out, poke into the water delivery port, and get the reward. So this is the sequence of actions. So it's a fixed set of actions, but the transition times when an animal decides they want to perform the task are self-initiated. And so what they found was that there's a huge variability in waiting times. And so what I'm showing you here, every vertical bar is one particular trial, and the dashed lines here are the waiting time for the impatient trials, and the full black lines are the waiting times for the patient trial. So you see that the variability just in the waiting times are among all the possible actions the animal is making in this experiment. There's this very, very large variability. And so the question we're going to ask in this experiment is, so first of all, how does the brain represent the intention to act? How does the brain represent the action in any one of these different steps in the experiment, or these syllables in the experiment? How then does the animal generate the action sequence? So how does this sequence of actions arise from a neural circuit? And in particular, I will also want to focus on how does the brain generate this variability in the waiting times, or in all of the other intervals that also have the same variability, even though I just showed you the variability in waiting time. So we have a stable behavioral sequence with very, very variable, with a large variability in the transition times. So the way we went about to answer this question was, so Mazzucato was recording neural activity from the secondary motor cortex of rats engaged in this task. So the secondary motor cortex was believed to be encoding for anticipatory activity. There's a large literature of that. And so what I'm showing you here is 3 raster plots, and so you're going to see a bunch of those. So a raster plot that represent the activity of 12 simultaneously recorded neurons from one ensemble, one session, during 3 trials in this task. And so every line here is a neuron. They go from 1 to 12. And every black mark is an action potential, or a spike emitted by that neuron in that particular trial. So this is trial 1, trial 2, trial 3. And these are trials where because the animal is performing the action at any given time, I just annotated every raster plot with an arrow describing when the animal was performing that particular action, or when the first tone was played. So poke in, tone, poke out, water poke in, and water reward. So, okay, so how do we go about analyzing this activity? So if you just stare at the raster plots, and there's like hundreds and hundreds of them, you can see that there's some kind of spatial temporal pattern of activity that occurs in every trial, even though it occurs at different time and for different duration. For example, I highlighted here one in red, the 3 boxes showing this activity close to poke out. And then, at other times, there's different patterns of activity. So I highlighted the second pattern of activity with the gray box, for example. In the gray box, you see that neurons 11 and 12 fire with high rate. The others are almost silent. And in this other red pattern of activity, you see that, in fact, neuron 12 goes down, and some other neuron comes up here, and this is what happens in all trials. So we use a standard way to analyze the spatiotemporal dynamics in an unsupervised way, and so this is called a hidden Markov model. So we fit the Poisson hidden Markov model to all the trials in the same session, and what I'm showing you here is the result of the fit. So the model is capturing, segmenting in an unsupervised way the activity in each trial according to some hidden states, and each hidden state is represented by a firing rate vector, a population firing rate vector. So we have 1, 2, 3, 4, 5, 6, 7, 8 hidden states in the first trial. So this is a sequence of hidden states. Within each state, the model assumes that the neurons are emitting spikes with the Poisson distribution conditional on the firing rate defined by that hidden state. And so what we see here, I can talk more about that. So there's a lot of details in the paper, if you like. So this is pretty standard technique. So the first thing we can see is that it seems like all of the sequence, there's the same sequence of hidden states, of patterns, if you want, in all of the 3 trials. And in fact, this is not a fluke, because if now we represent on the same plot all the trials in this session, the trial where the animal was performing this sequence of actions, every line here corresponds to one of these raster plots. So we have 3 raster plots here that correspond to 3 lines where we have color coded the same intervals that's on the left. And what you see is that across trials have different durations, and across all trials we have the same sequence of states. So what we did then to get a better sense of what the trials look like is that we stretched the trials to align them to some behavioral events so we can visualize them better. We can time warp them. And when you do that and you align the trials, stretching them to poke in, poke out, and water poke in, you see that the same sequence of states is present in all trials, and the same transitions between states are aligned to some of the behavioral events. And so now what we wanted to do at this point was figuring out, well, is this like the best session that we had, like that I'm showing you? In fact, no. So every animal, every session, we always find this structure of sequence of patterns, and it was pretty striking. So these are all the animals we have in the experiment. So this is the standard way secondary motor cortex circuits generate dynamics in this self-initiated trial experiment. And so the second feature of this, so the first feature is the stable behavior, stable actions sequence, and stable sequence of attractors that we find. The second feature, which is pretty striking, is that the duration of each attractor in each trial varies widely. So for example, if we look at the duration of this gray attractor here, this is like, it varies a lot from trial to trial. Even though it appears in all trials, its duration varies a lot. So if you look at the histogram of the duration of all patterns in all trials, we see that it's kind of approximately exponentially distributed. It's very skewed, with an average time of half a second. So these attractors are very long-lived compared to, single neuron times constants. So we hypothesize they might be generated by cortical emergent dynamics in this collective assembly of neurons. So the first question we want to ask is, what do attractors encode? So when we stretch this attractor sequence, we align them to these behavioral events, these 3 ones that I'm showing you here: poke in, poke out, and water poke in. We see that there's consistently onsets of attractors. For example, this red attractor, the onset consistently precedes the poke out transition in the behavioral sequence here that the animal is performing. And so we did is we tested the hypothesis that the onset of each attractor predicts an upcoming action. So what the animal is going to do next. And so what we did here, so I'm showing you here the trials, which are the correct trials, where the animal is performing the correct sequence of events, of actions. And below here, I'm showing you other trials where the animal was doing other stuff. So these animals are, their behavior is pretty wild. So sometimes there's a few, we call them error trials, where the animal are not performing the action in the correct sequence, but they're poking in and out many times and so on. And so what we did is we trained the dictionary between attractors and actions and onset of actions in the correct trials. And we used this dictionary to predict the actions the animal was performing in the error trials. And what we find, I'm not getting into the details of this, how we define this this decoder. It's in the paper, so but we can talk about it if you want to know more. So the accuracy of prediction, the cross-validated accuracy of prediction of this decoder in the correct trials was perfect, and we were able, using this decoder trained on the correct trials, to predict with very high accuracy the actions performed in the error trials. So we conclude from here that the way the brain represents action is that in the secondary motor cortex, we have patterns of neural activity, which we interpret as attractors, whose onset predicts what the animal is going to do next by, on average, 150 milliseconds. So the onset of every attractor here is going to predict what action the animal is going to take in the next 150 milliseconds. And so we have now characterized secondary motor cortex activity in terms of sequences of attractors that are reliable and stable, but whose variability in transition times is huge, is exponential. And so the question we want to ask now, because we like to do models of brain circuits, is what is the simplest brain circuit that can produce stable sequences of attractors, but where the transition times between an attractor is exponentially distributed, has this huge variability in waiting time. So this is what we're going to move now. We're going to switch gear, and we're going to think about models. Unless maybe I should pause here and see if there's any questions on the experiment.
Yeah, there is at least a few questions here, Luca. One is from Yuri Buzaki. He asks, in the hidden Markov model, how do you take into consideration the wide range of baseline and correlated task-dependent firing rates of neurons? Fast-firing neurons. Oops, missed the last part of that question. Okay, how do you take that into account?
So, okay, so how do we take into account the different, different cell types, for example? So the model is completely agnostic. Our data here is from extracellular recordings. So we don't have access to the cell type of the neurons. And so we are completely agnostic. We put all of the single units, that were spike sorted from each ensemble, and we just fit the model to the whole ensemble. So we didn't really think about cell types specific questions here, although that's a super interesting question, and hopefully we'll be able to answer that with optotagging maybe in future experiments.
Yeah, so actually Yuri was asking about, fast firing neurons tend to participate in multiple states and task phases. Do you have information about who's fast firing in a ground state or not fast firing?
So yeah, so I haven't thought about that, but I can answer the question about the participation. And so what we find is that, so we can ask the following question. So given a neuron, can we find out how many states modulate or the activity of that particular neuron? So a neuron can have the same firing rate across all states, so it can basically not participate in any state. That would be the bottom neuron here that's not modulated by the firing rate sequence. But some neuron can have modulations by one state, by several states. So can we quantify that? Can you quantify the quantity of neurons whose activity is modulated once, twice, or is modulated independently by all the attractors? And so what we found is that across all the neurons we recorded, we found that 90% of the neurons have activity that's modulated by the attractors. And let's say 50% of the neurons have at least 3 or more firing rates across these different attractors. So they have a very complex pattern of neural activity across the attractors. Although, yeah, so I can't say anything about fast spiking and not.
Okay, here's another question. This comes from postdoc Dan Levinstein, who just finished his PhD in the Buzaki lab. Does the exponential dwell time distribution come from the Markovian assumption in the HMM? I.e., would it be possible to fit an HMM and not get an exponential distribution?
Yeah, thank you. So, so that's a very good question. So, so the underlying assumption of an HMM is that you have an exponential distribution of duration, right? So, if you generate model from the HMM, that's what you get. If you fit back the model with the HMM from a model that's been generated by the HMM. If you fit an HMM to data, so the distribution of duration times that you get can be very, very different from exponential. So if you, for example, if you fit hidden Markov model data to evoked activity in sensory cortex that's evoked by particular stimuli, and then you look at the distribution of dwell times, you find that it's not like this. It's like it's modulated by the timescales that are present in the stimulus responses. So even though it's an assumption of the model, when you fit the model to data, you could get very different distributions from this one. Yes, that's a very good question. Thank you.
Very good. Thank you. Yeah. Okay, one more question, then we'll go on. Jonathan Cannon asks, has anyone looked at whether the basal ganglia and striatal dopamine play a role in initiating transitions among attractor states?
Thank you. Yeah, that's a phenomenal question. So in the seminal work that I mentioned at the top of the talk by Bob Dada, they analyzed the role of subcortical areas, including the ventral striatum and the basal ganglia in motor generation, in generating these self-initiated actions. And so there's a beautiful series of papers about that. We haven't looked at that. So although, as you can see, the model that I'm going to talk about now requires the presence of a subcortical area to generate this. And now with the Mainen lab, we have an ongoing series of experiments whose goal is to figure out, to test hypotheses as to which subcortical area is coordinating their activity with M2, with the secondary motor cortex, to generate this dynamics. That's a very active direction for us. Yes, thank you.
Okay, let's let you go on, and there's more questions. We'll hold them until a little bit later. Thanks.
Awesome, thank you. Okay, so now let's switch gear and let's talk about models now. So I want to model this neural dynamics that we found in secondary motor cortex in terms of a recurrent neural network. So the simplest neural network that everybody has in mind is a rate network, like the one I'm writing here, where UI is the activity of each unit in this network. This is like a recurrently connected, and Jij are the connectivity weights, the synaptic weights in this network. And so if we choose Jijs that are Hebbian, if you want, they are symmetric in some vectors, psi mu, and we have p vectors. This model is the standard continuous version of the Hopfield network that everybody is familiar with, where if we initialize the network in random initial conditions, then the network activity is gonna quickly end up in one of the attractors and just stay there. So this is like a model that generates stable attractors, p of them. And so of course, this is not what we want. We want a model that generates sequences of attractor. And so the simplest thing you can do is you can add a second term in the synaptic connectivity matrix that concatenates consecutive attractor. So if an attractor is a psi mu vector, then psi mu is the Hopfield term, and we have a second term that concatenates psi mu with the next one in the sequence, psi mu plus 1. So if we just simulate this simple model, we find that there's a phase regime in this model where the behavior, the dynamics depends on the relative strength of the symmetric and asymmetric part of, or we call it Phi4 part in the connectivity matrix. So for example, for very small values, there's nothing going on. The activity just dies out very quickly. For large symmetric part, the Hopfield network, we have stable attractors. This is what I just showed you in the previous slide. But then there's another regime of parameters where this concatenating part in the synaptic weight is very large. And then the model generates sequences of attractors. So this is promising. Okay, so let's see if this model works. Let's look at this. And so, okay, so in this model so far, there's no variability. So this is generating stable sequences, but there's no variability in the timing. So what we thought at the beginning was that, well, let's just introduce some private noise such that every neuron has some input white noise, for example, or Stein-Uhlenbeck, independent for each neuron. Does this work? And what we found is that this doesn't work. So this model with private noise generates very stable sequences of attractors, but they're too stable. So the variability in dwell times is very, very small. It's definitely not at all like the one that we saw in the data. And well, we thought, well, let's just crank it up. Let's just crank up the noise. It's going to work. But in fact, to our big surprise, Ulysses was analyzing the phase space of this model, and he found that no, there's no range of parameters of private noise independent for each neuron where this model works. So for small noise, you get sequences with no variability, very small variability. If you crank up the noise, you destroy the sequence at all. So these kind of models don't work at all. So why doesn't this work? And so the intuition behind this is the following. So we are in a very high dimensional space of neural activity. This is n dimensional, where n is like 10,000 for us, for example, like a local patch of the cortex. And so in order to be able to generate the sequence, and to have the transition between each attractor generated by noise. So here, this is like a cartoon of an energy landscape right. So where every potential wire represents an attractor, then we're here, and we have to jump here. But the jump must be generated by noise. In order to jump from here to here along a very specific trajectory, we have to pick that trajectory out of n possible direction, where n is very large, right? So there's no chance that we're going to do that consistently with random fluctuations, where every one of these n directions has an independent private noise. And so in the large n limit, there's a zero probability that you generate the transition in the right direction. So that's why this model never works. And so this is the curse of dimensionality. And so, but this also gives you an idea for how to make the model work. And the idea is that instead of having every neuron fluctuating independently, like we did in the previous model, we need to introduce correlated variability. So if you're here, we need to generate correlations that with fluctuations in the direction of the next attractor. And then you're going to generate a transition from this to this, and then from this to this, and so on and so forth. So what we do is that, if you remember, we had this feed-forward concatenating term that concatenated the sequences in our model. Instead of having an additive private noise, we add a multiplicative scalar noise in front of this feed-forward connectivity part of the connectivity metric, this asymmetric part. And so this is low-dimensional correlated noise. And when we do that, this was the breakthrough that allowed us to actually generate now stable attractor sequences. This is like a model simulation of the model, where you can see highlighted in different colors the overlaps with the attractors. And so the model goes through a stable attractor sequence, but the duration of each attractor now is exponentially distributed. So it's very, very skewed. And when we compare the model to the data now, the model really captures this stable yet temporally variable character kind of feature of the sequences of patterns in the data. Now, we want to test now some model predictions that were not baked into the model, right? So the first model prediction is that the noise correlations here are all the fluctuations are determined by this term, this asymmetric term that concatenates consecutive attractors. And so the noise correlations are going to be very low dimensional, because the dimension of the noise correlations now, or the fluctuations around each attractor, these are what I call noise correlation, they are bounded by the number of attractors, not the number of neurons. So they're at most p dimension, where p is much smaller than n. And in fact, they must be highly aligned between consecutive attractors, so that the fluctuations in this well is very aligned with the fluctuations in this well, and not as much with the fluctuations as you move away in the sequence. So we have two predictions. Let's say low dimensional noise correlations and highly aligned noise correlations. And so we went back to the data and we compared these two predictions in the model and in the data. And what we found is that, first of all, in the model, the dimensionality of the noise correlation now are the fluctuations conditioned on each attractor, are very low. And when we look in principal component space, if you wanted the data, now the noise correlation is the dimensionality of this cloud of points conditioned on the red attractor here in the data. We find that these patterns are very low dimensionals, at very low dimensional noise correlation, just like in the data in principal component space. And in fact, the most striking prediction here was that the noise correlation between consecutive states in the sequence, for example, here the gray one and the red one, should be aligned. So we form a canonical correlation analysis, and we found, in fact, that the cosine of the angle, if you want, of the canonical correlation between patterns that are consecutive is very large. But as you step away in the sequence, the canonical correlation goes down in the model, just as like in the data. And so these two predictions of low dimensional, highly aligned correlations are confirmed in the data. So let me wrap this up, the first part of the talk. So I showed you that upcoming actions are represented by metastable attractors in the secondary motor cortex. And these sequences are concatenated by low-dimensional correlated variability. So although this model is a phenomenological model, in the paper, if you want to know more about this, we show how this model arises from a recurrent loop between the cortical circuit, that's the secondary motor cortex, and a subcortical area where this noise is arising from. So more details are in the paper. So why do we have this timing variability? What's the interpretation? Why is this useful to have stable behavioral sequences with variable timing? That's because okay, so this is a possible interpretation, is that, you know, when you have to perform very complex sequence of actions with perfect timing, you get beautiful videos on YouTube, of course, and in particular, you know, many animals are very good at performing complex behavioral sequences with perfect timing. But how do they get there? Well, to get there, you have to be able to experiment and have a variable timing. You know, because the first 100 times maybe you get it wrong. You know the sequence of actions you have to perform. You have to eat the fish, right? But you just get the timing wrong. And so maybe this is a good way to experiment with timing with this variable duration of attractors until you learn the right timing for a sequence. So this is a possible interpretation for what we have. Okay, so. Okay, let me pause here and see if people have other questions. Okay, if there's no other questions on this part.
Unmute myself.
Oh yeah, please go ahead.
Arrow asks, is there any correlation in the duration of the different intervals between one trial, i.e. as if conditioned on some master speed control signal?
Yeah, so the model that I presented to you is Markovian in the sense that these the transitions are independent of each other. And to the first approximation, that's what we found in the data, is that there's to the first approximation there's very little correlation. However, there is some correlation between the waiting times in different trials, for example, and as the waiting time in trial t depends on the waiting time in trials t-1 and t-2. So there's a small amount of variance. Oh, yes.
Go ahead. Yeah, go ahead. Sorry.
Yeah, there's a small amount of variance in the durations of sequences in one trial, depending on the previous trial. That was discussed by Zach and Mazza in one of the papers. And so to account for that, we need to go beyond this model. So this model captures, if you want, a large amount of variance in this task, but there's a little subleading effect that's not accounted for by this model. And so we need a longer timescale. We need to add some extra ingredient in this model to have a longer timescale, likely arising from projections from other areas. So, for example, for the median prefrontal cortex, to account for this longer timescale. So that's what we're currently thinking about. Actually, that's a very good question. Yes.
Okay, we'll come back to more questions a little bit later. Why don't you go ahead? It's now 12:48. So normally we would stop in 15, 20 minutes from now.
Oh, that's great. Okay, so I have time to talk about a little bit about two other projects. Okay, the next question we're going to ask is like, okay, now we have this interpretation of cortical activity in terms of metastable attractors, right? So, but you want to be able to flexibly control the timing of these attractors, or the onset of the attractors, to adapt behavior to different contexts, for example. So how do neural circuits control their processing speed? And so the idea that I showed you so far is that, we have these attractors that put as, like, let's say, potential wells, and then the transition times from one attractor to the other attractor depends here. So I put a schematics here on the left. Depends on the height of the barrier separating the attractor, right? If this is a diffusion process, the leading term in determining transition time is the height of the barrier. So now I want to ask the question of how can neural circuits control these transition times between attractors? So now we switch perspective and we think about sensory cortex, and we think about the onset of attractors as stimulus responses in sensory cortex. So we have a model of cortical circuits in terms of, so it's going to be a different and more complicated model that's more biologically plausible in terms of excitatory and inhibitory spiking neurons. And so these kind of models where neurons are arranged into cortical functional assemblies or clusters, if you want, generates metastable dynamics. So what I'm showing you here is like a snapshot of this biologically plausible model, where this is the synaptic weight matrix, where neurons are arranged in clusters. You see these diagonal blocks of different sizes. And here I arranged neurons in, I plotted neurons according to the cluster membership, and you see that these clusters become active and inactive at different times. So this is like spontaneous switching dynamics between attractors that's characterized by an intrinsic timescale of this cluster activation, if you want, which we call tau. And so we can summarize the activity in this network as some kind of during ongoing periods where there's no external stimulation, as switching, a cluster switching on and off with a certain particular intrinsic timescale. So now we want to be able to classify the effect of perturbations that encode context dependence from, for example, top-down projections from frontal areas to sensory cortex, or neuromodulation from subcortical nuclei into the sensory cortex. How do perturbations change the dynamics of these switching dynamics of these networks? And so, for example, one perturbation we can think about is modeled in the following way. Every neuron here receives a constant input current. That's the baseline input current. Let's say we start with the same value for all neurons, and then we just modulate the value, the mean value of this current as for each neuron as a dial. We can dial it up or down, and we can change the mean across neurons if we all change it in the same way. Or here, what I showed is that we can change the variance across neurons. So we change the input, the baseline input to each neuron in a different way, such that the mean is 0, but we only introduce some spatial variance across neurons. So this is one particular perturbation, and we can classify all the possible external perturbation to the network, and we can study the effect on this intrinsic timescale, on these attractor switches, that each of those perturbations is bringing onto the network once it's online. And so in this particular case, we found that in introducing this increasing external variance to the network shrinks the barrier between the two attractors. And so it's introducing a faster switching time. So we can control the timing of these ongoing switches with these external perturbations. And so now we want to test this theory experimentally. We want to go into a circuit and see whether it changes in barriers of in the barriers between attractors control the timing, the intrinsic time of this neural circuit. So however, we can't possibly be able to measure, energy potentials in neural circuits. So how do we go about to test this theory? And so using some theoretical tools like mean field theory, one can show that the height of the barrier between attractors is equivalent to the gain of the single cell transfer function. So there's an approximate relation between a potential, the potential energy in this picture, and the integral of the transfer function, so that neurons that have a steeper gain have a higher barrier, and neurons that have a milder gain have a lower barrier. So by measuring gain modulation now, we can predict whether the activity is going to speed up or slow down. For example, if we find a modulation or a perturbation that lowers the gain, this theory predicts that that's going to lead to faster transition times, just like here. And so we go about to test that. And in particular, if we before testing that, let me add that now this was during ongoing dynamics, and now we want to predict what happens during stimulus evoked activity. So when we present a stimulus to the network, let's say a visual stimulus to visual cortex, that's gonna have some selectivity. So some of the clusters are the stimulus selective clusters. They have some onset latency to get turned on, right? So this is after stimulus presentation, the selective clusters are gonna turn on with some latency, and the perturbation is gonna change this onset latency as predicted by the theory. If it's a perturbation that lowers the barrier, then we're gonna lead to a faster onset time. So a perturbation, an external perturbation to the cortex, to the cortical circuit that lowers the gain of neurons, according to the theory, predicts a faster response to stimulus. So what we did is that we went about to test this, and this was work done by David Wyrick, a student in the lab. We used the Allen NeuroPixel dataset to test this theory, and we looked at recordings from with NeuroPixels from all the visual cortical hierarchy in the mouse. And so what we did is we first measure whether okay, so what's the perturbation we're thinking about here? The perturbation that we're focusing on is locomotion. That's the simplest perturbation we can think about. So the animal is going to be running or resting, and these are the two contexts that we're gonna measure. And so we're gonna look for a change of gain in the single neuron transfer function that's generated by running. So if single neurons during running have a different gain, the single neuron during resting, and then given that change of gain, that's going to predict the effect of running on the latency of stimulus coding. And so what we found was that in all the visual hierarchy, these are the 5 visual areas in this cortical hierarchy, there's a decrease in gain during darkness. So during darkness periods, the animal is running in the dark. Still, we measure neural activity. We can estimate the single neuron transfer function. And we saw that in darkness, when the animal runs, neurons have a lower gain than when the animal is resting. So according to the theory, this predicts that running, when we now present stimuli, running is going to accelerate stimulus coding. This is what the model predicts. It's an acceleration of stimulus coding in brown compared to the black curve. And this is what we found in the data. We found that there's a significant acceleration in coding in all the visual areas, just as predicted by the model. So this was like a test of our theory. The gain modulation induced by perturbations controlled the processing speed of the circuit, and this was confirmed with running. And the previous work that I did when I was a postdoc at Stony Brook, I showed that the same effect is present also in gustatory cortex, now a different cortical area with the taste stimuli, and it's triggered by expectation of taste. So, which is another possible context dependent perturbation. And so in the bioarchive, you can see in the paper, we classify all possible perturbations to the circuit. And I want to give a plug to some cool work that we did in the lab with Audra McNamee, who's a comic artist. We drew a graphic novel on neuromodulation. So this is all work about, this is motivated by studying neuromodulation. So we wrote a short graphic novel on the effect of serotonin on cortical circuits that, if you're interested, you can find on the lab website. And so it's very cool art by Audra. Okay, so that was the second part of the talk, and I can pause for questions, and then I can briefly go through and wrap up with the last part of the talk.
I think we should go ahead and wrap up, and then we'll take questions at the end. How about that?
Okay, so if I can take 5 extra minutes, I want to talk about now how to put together all these ideas. So at the top, I showed you that in these YouTube videos, we see animals having a very large hierarchy of timescales over several orders of magnitude emerging in behavior. So how can we put together networks with this that robustly generate this huge hierarchy of timescale? And of course, this work was motivated by this amazing series of papers by Xiaojing, where Xiaojing and collaborators studied the emergence of this hierarchy of timescales across cortical areas. So what Xiaojing found was that across the cortical areas from the posterior to the anterior cortical areas, there's a increase in intrinsic timescales in the average population dynamics of these cortical areas across a pretty large span of different timescales. So this is what motivated it. So, but the question we're asking now is a slightly different one, is how does this hierarchy emerge within the same cortical circuit? So at the level of single cell autocorrelation. So what was found was that not only is there a hierarchy between cortical areas, but within the same cortical circuit. For example, here, this is from the Johnny Wallis lab, from anterior singular cortex, the neurons in the same circuit have a huge hierarchy of timescales. And so the question we asked was, what's the simplest circuit that can generate these multiple timescales over orders of magnitude within the same cortical recurrent network? And so the simplest thing we can think about is a mean field model, where every unit in the model now, this is the same model as before, every unit in the model represents a functional assembly, a neural assembly. Here, for example, we have 3 assemblies. They are like green, blue, and red. And some assemblies that have some random recurrent connectivity, JJ, plus a term that represents the self-coupling of each neuron. That's like the strength of each cortical assembly. And this network have a very cool chaotic dynamics, and that was found by it was studied by Markov in a previous paper, our collaborator. And so in this simple network, however, if all neurons have the same size, if all cortical if all units that represent cortical assemblies have the same size S, then this is represented in this synaptic weight matrix. The diagonal is this self-coupling term. The off diagonal is just a random Gaussian ensemble. In this network, there's no heterogeneity of timescales at all. So all neurons have the same intrinsic timescale. That's determined by S and the variance of the diagonal term. So this is not enough. And so what we thought was, well, let's introduce a little bit of a biologically plausible ingredient. Let's now consider assemblies of different sizes. So how do we introduce different sizes, heterogeneity in the size of the cell assemblies? Well, just by having different values of the self-couplings. And so, for example, the simplest one is where we have half of the assemblies in the model have one value of self-coupling, and the other half have a different value of the self-coupling. And what we found is that, yes, so this is enough to generate different timescales in the model where the timescale now, this is the autocorrelation of neurons in one assembly and the autocorrelation of neurons in the other assembly. The timescales are determined by the size of the assemblies, the self-coupling term. And so as we consider larger and larger separations in the self-coupling, we get larger and larger differences in this timing. And in fact, if we choose, for example, a log-normal distribution of these sizes of cell assemblies, we get a huge variability of autocorrelation times, which in fact increases exponentially with the value of the self-couplings. So we get self-couplings varying over 3 orders of magnitude easily in this model with log-normal distribution of self-couplings. And so, okay, so that's it. Let me wrap this up. So, so what we found is that, okay, so we have a proposal for how stable action sequences with variable timing is generated in the brain. So every action is represented by a metastable attractor in secondary motor cortex, and the variable timing in this concatenated sequence arises from this low-dimensional correlated variability, which, you know, in terms of information limiting correlation is detrimental for sensory coding, but it's necessary for motor generation, which is kind of a new point of view on this low-dimensional correlated variability. Then we show that we can control the timing of these transitions with external perturbations, and we can classify that using gain modulation. So the effect of perturbations that control the timing are encoded into this gain modulation that we can test. And then I showed you that this huge hierarchy of scales now within the same circuit can arise by the heterogeneous cell assemblies. So to conclude, I want to remind you who did the work. So the first part of the talk by Ulysses, who's here in Xiaojing 1 slide now, and Stefano with Zach and Masa and Champalimaud. The controlling processing speed, it was done by David Warwick, who's a grad student in the lab. And the last part I talked about this hierarchy of timescales in these recurrent networks was done by Merav Stern and Niku Istrate. And all of the work has been debugged by my cat, so don't believe anything I'm saying. And so I wanted to take the last 30 seconds to plug. So if you like this work, we have an ongoing collaboration with Rusbekiani, where we are now testing these ideas of metastable dynamics using closed-loop brain-machine interface, where monkeys are engaged in a decision-making task, and we monitor and stimulate neural activity from prefrontal areas to trigger stimulation on particular brain states, the ones I talked about, to control the animal's behavior. So we're looking for grad students or postdocs. So if some of you is interested, please contact Rusbe or myself. And I think I can stop here.
Well, Luca, thank you very much. Appreciate your presentation in this set of projects. We have a few questions here. One is by Dima Rinberg. He's asking, you're talking about the transition in neural states, which can be correlated directly with behavioral events or stimuli. The earlier works observed similar transition just during internal stimulus processing and decision-making. a la Miller and Katz 2010 on state transitions in gustatory decision-making. Can you comment on this phenomena? Do we actually need behavioral manifestation of state transitions?
Thank you. Yes. So I'm so glad you brought this up because I was as a postdoc, I was working exactly on that kind of data that you mentioned. This data from the taste cortex, from the Fontanini lab at Stony Brook, and was very much informed by the work from Don Katz at Brandeis. And so in that case, we showed that there's state transitions, and so Miller and Katz that you brought up showed that there's state transition during stimulus-evoked activity. Those are similar to the ones I showed in visual cortex, for example. But what we showed was there's also, with Giancarlo Camera, we showed that there's also state transitions during ongoing activity that don't have clear behavioral readouts. So it's definitely not necessary to have behavioral readouts to have state transitions. And so, for example, if you have state transitions in sensory cortex, but the downstream is gated, so this is not read out, these transitions are read out, so they're not going to be behaviorally relevant, even though they are the intrinsic dynamics of this particular circuit, or they might be, in some cases, like in visual cortex, they might be driven by the backdrop of movements, of proprioception, somehow, like the Carandini work showed, the Stringer work showed. So it's a very complicated story, and it depends on cortical areas, and we just gave a couple of examples of where they could be relevant. Yes.
Okay, thanks. Another question here is Yuri Uzaki has another question. State variability in the waiting time figure he's asking about. If you plot the data on a log scale, you may see a very nice log normal distribution of waiting times.
Yes, thank you. So, in fact, it's hard for us to tell the exact distribution of waiting times. It could very well be log normal. So I agree with that. In some cases, there's a huge variability in the shape of the distribution between attractors and between sessions and between animals. So that the ones I showed kind of looks like in between exponential and log normal, I would say. So it's hard to tell really, but yeah.
So I have a question. So we have this sequence of stable states that we're progressing through in this sequence, and it's noise driven from one element to the next in the sequence. How do you control the noise so that you don't go backwards or you don't jump back, or if you wish to restart at some point, how do you do that in some intelligent fashion or unintelligent fashion?
Yeah, so that's a very good question. So in the model that I showed, there's no way to do that. So it's a model that explains that particular behavioral sequence that the animal has learned, right? So, but what we would like to think about is this kind of question. So how can you use noise? How can you learn to use noise to change, to switch between behavioral sequences and to go backwards, for example? So I don't know how. There's been kind of different series of papers on similar questions by Laureline Lojaco, for example, at Columbia with Larry Abbott and Sean Escola. They study how using a model that includes some other subcortical nuclei, like the basal ganglia, you can switch cortical sequence. So that's a model with cortex, thalamus, and the basal ganglia, and it's generating switching between behavioral sequences. So we're thinking about that, but I don't have any answer today.
Okay, we have anybody else have a question? If not, I think we can close shop. And I can't tell you when the next Swartz seminar is. I don't know at this moment, but stay tuned. And Luca, thanks again for a wonderful and fun talk. Thank you.
Thank you. Thanks for the great questions. Thank you. It's been fun.
Okay. Great. Have a good day here.